As I said, this is Genetics Beyond the Basics with Dr. Melissa Crenshaw. Dr. Crenshaw is the medical director for the clinical genetics program at Johns Hopkins Children's Hospital, as well as a new promotion to associate professor with Johns Hopkins Medicine. Dr. Crenshaw focuses on the genetics of congenital heart disease and connective tissue disorders. Please all welcome Dr. Melissa Crenshaw.
Thank you so much. It's a pleasure to see you all here today and join us for the afternoon of genetics. And we'll have lots of time for questions at the end. So we will, and also we'll be sending them in on the VVOX poll. Now this is my first time using the polls, so bear with me. But we're going to talk a little bit about what makes us who we are. So genetics, beyond the basics. So we'll start with a little bit of the basics and get everybody on the same page. And then we'll talk a little bit more about some of the nitty-gritty. So let's start with a little bit of perspective. Why is our genetics important? So what is it that genetics does for us and helps us? Each of us has a different journey, so we'll talk a little bit about that. I'll tell you a little bit about my journey as well along the way. We'll talk about what makes us each different and what does different mean to us. And that's different for everybody. So looking around, we could see from that picture that there were a lot of differences. Different hair colors, different smiles, different skin colors, and different forms of dress. So we're all unique. And what makes us different is a combination of genetics and environment, right? So our genetic blueprint makes us different, but also a lot of where we grew up, how we grow up, differences in values and priorities. Different doesn't mean abnormal. So we're all different from each other, but that doesn't mean that that difference is abnormal. And not all genetic changes are visible. So this is just a quick picture to kind of give you an overview of genetics. So all of our genetic material is in the center of the cell, in the nucleus. And our blueprint, which is housed in our chromosomes, tells our body to make proteins, which are the building blocks. It tells our body how to be put together when we're developing. And differences in that blueprint lead to differences in that process. So our genetic blueprint, what makes that up? Well, we have 20,000 genes in our cells. So of course, they're not all on the X chromosome. They're not all involved with Turner syndrome. That's only a small portion. So all of those 20,000 genes make us who we are. And they're in the nucleus of the cell. We call that the little control center. And we could think of this as our library, where the books are housed. We have the bookshelves, and those are the chromosomes, and the books are shelved on there. And we share 50% of our genes with our parents and siblings. So we know that we're more alike from our family members than not. But also have a lot of differences. So we have 23 pairs of chromosomes, 46 total, 22 are the same for men and women, and then the 23rd pair is the X chromosome and Y chromosome, and those are our sex chromosomes. So 2X for women and an X and a Y for men. Can we get back to our slide here? Okay. And that 23rd pair, as we said, our females have 2 X's and males have an X and a Y. And how do we get this library? How do we figure out who has 2 X chromosomes and who has 1 X and 1 Y chromosome? Well, each of our parents contributes 1 copy, right? So when we're conceived, each parent contributes 1 copy, and that's true for each of our pairs of chromosomes. So the egg and sperm cells come together, and our dad decides if we're male or female because he contributes either an X or a Y. So dad gets to figure that out. So going a little bit closer are the genes. These are the books. So we talked about the bookshelves, which are the chromosomes. So the genes are the books. They tell our specific plan. So the stories in those books tell how our body develops and how our body will progress through life, whether we'll develop issues with hearing, for instance, or issues with high blood pressure. They tell our specific plan, but each of us has a different story because every one of us has very different genetic material. And difference what makes us special, right? So even regardless of X and Y chromosome, we're different, and that's what makes us special. We could be missing books or have extra books, and the books can be spelled differently, right? So what's a karyotype? We hear a lot about karyotype. What's a karyotype? So it's a picture of our chromosomes. It enables us to count the chromosomes under the microscope. It looks for big changes and rearrangements, and it's how we diagnose Turner syndrome most often. So that's the common test we use to diagnose Turner syndrome. So to truly know if somebody has it, we have to do a chromosome test. How many of you know your karyotype? And we'll stay tuned for our next talk, where we'll talk in more detail about different karyotypes. So this is a typical karyotype here, and this is just a cartoon of it. But you can see there's lots of little bands, and that's how we tell if all the material is there and if all the chromosomes are there. They each have their own unique pattern. So in Turner syndrome, females are missing all or part of their second sex chromosome. And the specific karyotype could tell us more about specific medical complications that we need to pay more attention to. And again, we'll talk about that in more detail with the next talk. This happens randomly, so it's not something that causes this to happen. There's nobody that makes Turner syndrome happen. There's no medication that causes it or exposure during pregnancy. It's just a random event. And it can happen from either the egg or the sperm cell. So we don't know if it happens from the cell we get from our dad or the cell we get from our mom, and it doesn't make a difference. So this is just a quick picture of how that happens, that process that we call non-disjunction. So as you can see, there's two cells there. One has only one chromosome, and one has two chromosomes. One is from a different pair. And the pair to keep your eye on is the blue one, which is the X. And you can see that there's no second X chromosome. So one cell is contributing only one copy, and the other cell is contributing no copy. So you end up with just one X chromosome. And this is a picture of a couple of different Turner syndrome karyotypes, actually under the microscope. And the first one is an individual is missing an entire X chromosome. And the second is an individual that's missing a little piece of the X chromosome. So you can see on the right, those two copies of the X chromosome don't look exactly alike. They're supposed to, but they don't. So why is the X so important? Why does this mean so much? Why does it cause Turner syndrome when we don't have two copies? And why can men have only one and women need two? How does that make sense? So we'll talk a little bit about what does the X do and what does the Y do. They have a little bit different jobs. And then we'll talk about genes, because we have to do that to understand why this is important. So here's the X chromosome, the star of our show here. And that has about 80 to 1,000 genes. So not the most in our genome, but a good number. About 5% of all of our genetic material is in the X chromosome. And we know that in each cell, one copy is turned off, right? So one copy of the X gets to stay active, one copy is turned off, and that's random too. Nothing that specifically causes that to happen in a certain way. But it's not the whole thing. It's about 98% of the X is turned off, but about 2% doesn't get to be turned off. And that's called the PAR region. We'll talk about what PAR region means. So remember that part doesn't go to sleep. That part stays active on both copies of the X. Now the Y chromosome. So there's a few average genes there, and then we have SRY. That's called the testis determining factor. It's expressed for a very short period in development, so only during a certain period during baby growing. Those genes build the male aspects, such as sperm. They make a male a male. And a male produces billions and billions of those from puberty. And the main job of the Y is to bring that genetic material to the egg. So that's what the Y does. It gets that job done. But there is a PAR region as well, which is about 5% of the Y chromosome. And that's important too, because the PAR region is similar on the X and the Y. And so these are areas, this is just a little picture of where they are, and I'm not going to go into a lot of minute details about that, but this is a picture of where those PAR regions are. And so there are areas on the X and the Y that match up, right? Because our chromosomes have to pair when our cells are dividing, our chromosomes have to pair up, and that's how we get the X and the Y to pair up. But those are also genes that we have to have two active copies of, whether we're a male or a female. So the PAR1 has 22 genes, and the PAR2 has 5 genes. And like I said, we need two active copies of those, and it allows us to make sure that the sex chromosomes travel together in development. So genes on the X chromosome, we're still learning more and more about what all of those genes do. We don't know all those answers yet. We do know a lot about one gene, and that's called SHOX. And SHOX is the gene that causes the majority of growth issues in Turner syndrome and could cause the bones to develop differently. There are genes on the X chromosomes that are on the X chromosome that are involved with nonverbal learning issues, recognition of facial features, and use of social skills. And there's many more genes that affect the thyroid. So where are the genes? So this is just a little picture to show you where they are. So there's SHOX, nonverbal learning disability, stature and ovarian failure, minor physical features, prenatal viability. So we'll talk a little more about this one. Ovarian failure. So there's multiple genes on the long arm, which is Q. Q means long arm, that can cause ovarian failure. Genes that affect facial expression, recognition. So let's talk about SHOX. So SHOX accounts for about 2 to 7% of short stature in the general population. So some people can have changes just in that gene and have short stature. It causes about 2/3 of the height decrease in Turner syndrome. So it takes the most of the responsibility for the growth differences. It also causes a high narrow palate and can cause that change in the arms called Madelung anomaly that affect people with Turner syndrome. Not everybody has it, but it can cause that. The RPS4XY gene, I just mentioned, is associated with viability. So we know that not every baby with Turner syndrome comes to be born. So this gene, we think, is part of the reason why that happens. And that gene's on the X and the Y. And you have to have 2 copies of that active to ensure prenatal viability. It's part of the structure of the ribosome. And a ribosome is that little factory that makes proteins, okay? And without 2 copies, the embryo doesn't make proteins as well. Doesn't mean it can't, but it's not going to make proteins as well as an average developing embryo. So they think that the incidence at a baby's conception, when a baby starts developing, is 1 in 50, but we know that at birth, about 1 in 2,500 girls will have Turner syndrome. And this gene is probably the reason why that happens. So we think about 99% don't come to birth. So we talked a little bit about cell division, and Turner syndrome can happen at 2 different parts of cell division. So meiosis is what we talked about a little bit before with egg and sperm cells. It may not happen 100% as expected, right? So it doesn't always go exactly with the chromosomes dividing as they should. The karyotype can change randomly at that time, and when it does, that's in all of the cells. So in this case, all of the cells would be missing the second sex chromosome or a part of the second sex chromosome. And each of the cells has 1 sex chromosome, X or Y, when this happens. And and if there's non-disjunction or it doesn't happen properly, then cells can have no sex chromosome to pass on. Mitosis. So mitosis is a time where the cell divisions happen later. Mitosis is the cell divisions that happen after conception, right? So this is when that second sex chromosome may be lost in the process later on. And so that happens later in development. It happens after conception, and the karyotype can change randomly in some cells, but not all. So this is where we see a karyotype in only some of the cells and other cells having a different karyotype. And that's called mosaicism. So we talked about non-disjunction, and that again is when an egg or sperm cell ends up with 2 or 0 sex chromosomes. If an egg or sperm with an X pairs with the one that has no sex chromosomes, the baby will have Turner syndrome. This can occur in mitosis, and that can cause mosaicism. But other karyotypes can be more complex. So this is a general way we talk about how the karyotypes can occur, but we'll talk more about what each different type of karyotype is in the next lecture. So, as we can see from talking about all of these different times in cell division and different types of ways that this can happen, Turner syndrome can have many complications, but everyone with Turner syndrome is different. Not everybody experiences the same medical issues or the same complications. We're learning more and more about why that happens, why each of us with Turner syndrome are different from each other. There'll be more to come, like I said, in the karyotype talk. One of the other reasons for this is that our genes interact with each other. So we've got 20,000 genes, so we know that there's about 5% of the genome is in the X, but that's interacting with that other 95%. So not everybody's X interacts in the same way with that other 95%, which makes each of us different. The more we understand this, the better that we can predict complications and the better we can care for girls and women with Turner syndrome. So that's important. The more we understand these genetic differences, the more we can pick out how we may help a certain person with a certain karyotype versus other things we need to be helping somebody else with a different karyotype. This picture is a picture of the many faces of individuals with Turner syndrome. So as you can see, that looks pretty different from each other, just like that first picture, people look very different from each other. So even though many of us have Turner syndrome, we don't all look alike. We don't all have the same experiences and the same medical problems. And that's, again, because we have so much other genetic background that are interacting with that. So this is a great picture of different stages in life with Turner syndrome as well. You can see we have young and older people. So tell us a little bit about, in our next poll question, how does it feel to be different? In one word, how does it feel to be different from your friends or different from, as a person with Turner syndrome, how does it feel to be a little bit different from other people that you meet? There we go. So I think we have some great responses here. So having Turner syndrome, being different from others, as I have experienced myself, has a lot of different emotions, and they can be different through different times of life. But I think these are all really great ways of experiencing or explaining some of those feelings and voicing them. And one great thing that we have here is meeting other people with Turner syndrome. You probably share a lot of those same feelings with each other, and you can see from in the audience, there's probably some that you feel, and especially unique seems to be the biggest one that we feel, which is great. Okay, so the next poll is parents have a lot of emotions too. So I know we have some parents in the room that have children with Turner syndrome, and I know that when I was diagnosed at 12, my parents had a lot of emotions, and that changed over time. Their emotions also changed, and how they felt about having a child with Turner syndrome. So our next poll is tell us a little bit about how that feels as a parent. Tell us some of the things that about your daughter with Turner syndrome that, in one word, how you would describe. There. So I love it. The biggest one they have there is blessed. And Turner syndrome truly is a blessing. Again, some people have other feelings as well, and those are all very valid. And parents go through a lot of very different emotions over time, having children with Turner syndrome. But what we see here is a lot of love, for sure. So some takeaways are, one, genetics and environment work together to shape who we are. Our genetic blueprint makes each of us different and special. And everyone has changes in their blueprint. So we're not all alike, even if we don't have Turner syndrome. No two of us are alike. That's what makes us great, right? Everyone, or I'd say nobody causes our genetics to be different. Not our mom, not our dad. These are just random events. And we're learning more and more about how our genetic story makes all of us with Turner syndrome different from each other and how those genes interact. And we'll talk a little bit more in the next lecture about why some of those changes in the karyotype will help with medical care down the road. So thanks so much for your attention. I appreciate your patience through some of the technical glitches. And we'll open up for some questions. I want to leave plenty of time for questions. I know you'll probably have a lot. So please join us with your questions through the VBACs. Yeah.
If you take your phone and you look at the front page, you'll see a little chat icon, and you can go ahead and type your questions right in the chat. That way we can try to get to as many of duplicate questions as you have. And if that doesn't work, we'll do regular hands.
Mm-hmm. Yes. Mm-hmm. Right. Mhm.
Still don't think of buckets. We actually got it because of an ET test or something different. It popped. So how does that like affect really like knowing how we don't even fit in like the basic buckets? Like how do we really understand how our daughters are impacted, ourselves are impacted? Because it kind of feels like we don't fit.
I see. So you feel like features don't fit with exactly with the reasons for testing that we would average.
Right.
So,
I think that one issue is like we would have never been tested,
mhm.
and so we were tracking a totally different worrisome puberty
Mhm.
issue on our med, right? And then,
Right.
but now that we do know,
Mhm.
there's all these things that we're talking about with the learning disabilities and fertility and kidney. I mean, all the, I'm not in the medical field,
Right.
so all the things. And then it's like, well, that applies to this, that applies to this. Like, how do we like kind of build those like buckets to kind of understand how this data that we have now from our doctors can help us understand what to look for, what to ask about, what to coach our pediatricians about.
Right.
Both of them doing nothing. So
Right, and that's a great question. That goes back a little bit to how everybody with Turner syndrome is different, so not everyone checks all the boxes, right? But when we know there's that genetic change, we kind of observe for all of those different things. We don't say that those things will happen, but we observe for those different things. There are certain pieces that have to be there to make a diagnosis of Turner syndrome. Most importantly, the karyotype has to be one that's very specific to Turner syndrome, and we'll talk about that in the next as well. But not everybody will check all the boxes, and so that can be sometimes confusing because not everybody has, as typical features as others do. So I'll take a couple from the chat here. Have you noticed red hair being prominent in people with Turner syndrome? So that's a great question. That is not something known to be prominent specifically. And hair color does not seem to change with Turner syndrome, but that's a great question. But as yet, we have not seen a connection there. How often should a TS girl see a geneticist? That one is a really good question as well, and it depends a little bit on where and how you get your care. So if you go to a multidisciplinary clinic, most often you'll see a geneticist yearly because the geneticist often is the coordinator for the clinic or the endocrinologist is. So you may see a geneticist once a year. You may not need to see them as often. It may be every couple of years or intermittently. It really depends on each individual's care needs. There's not sort of a specific guideline for how often, but we're always there when you need us. What are your thoughts about MTHFR gene mutation in Turner syndrome? That's another good question. There's a lot of information out there about MTHFR that can be very confusing. There hasn't been any specific data or specific issue with MTHFR related to Turner syndrome or autism or learning problems for that matter. It's not been strictly connected to those things, and also variations in MTHFR are super common in the population, so they can be very hard to interpret as causing issues. Why are my daughter's eyes green when mine are brown and my wife's are blue? So that's a great question as well. So sometimes, genetics of eye color are a little separate from the X chromosome, but of course are a mix of those genes in the family. Brown is usually dominant, whereas green and blue are more recessive. So you kind of have to look back in your family at different eye colors. But it can be a super interesting thing to follow. Yes?
Along with the eye color, I, with Turner syndrome, have brown, but my parents both have hazel.
Mm-hmm.
So I've always understood that brown is dominant. So I always thought that was interesting that it went that way instead of flipped.
Mm-hmm.
But is there anything you can say about that?
Not really. That is very interesting. And there's probably some of that genetics we don't entirely have sorted out yet. But that is a very interesting pattern. Can you speak more on differences between classic versus mosaic? I was told that mosaic meant less symptoms and effects, but that's not been the case. So that's a really good question. And actually, we're going to get into that a lot more in the next lecture where we talk about the individual karyotypes. But that being said, the 45X karyotype isn't always more severe features than 45X mosaic with 46XX, for instance. So it doesn't always follow exactly. With mosaicism, there tends to be milder features, but that's not always the case. Can you reduce a karyotype as an adult? Or redo a karyotype as an adult, sorry. So yes, some people do have a karyotype redone if they've never really had one officially or they don't have the report. Some people do have them redone later in life, but one thing to keep in mind is that as we get older, our cells in mitosis do lose their second sex chromosome often. So women of a certain age will have a certain level of 45X cells in their body. So we keep in mind that the level of those, the cutoff gets a little bit higher as you get older to say that it's significant. I have twins. How is it that one has Turner syndrome and one doesn't? That's another great question. So twins, they're either fraternal or identical, right? So twins don't always have the same genetics if they're fraternal. So a twin can have Turner syndrome, whereas the other twin doesn't if they happen to be fraternal. And it's the same idea with fraternal twins. It's the same idea as two siblings that are born at different times. They share 50% of their genetics and 50% that are different. Is classic and mosaic Turner syndrome the only two variations? And so know that they're not. Classic Turner syndrome is the one we typically think of, and then mosaic is at the 45X, as we just mentioned, with 46XX, but there's multiple different karyotypes that can occur in Turner syndrome. So those aren't the only two. And we'll talk about in a lot more detail in the next lecture as well. Do you know if there's a link between Ehlers-Danlos syndrome and Turner syndrome? So there's not a known link between the two. They're caused by very different genetic issues. Ehlers-Danlos is caused by mutations in specific genes. There's at least 7, 8 or more different types of Ehlers-Danlos syndrome. And so it's a separate connective tissue problem. My daughter is 45X, but her karyotype, but one cell was 46XX, but I recall being told she's not mosaic. So that's a good question. I'd have to see the report to know, but usually before they'll call a certain number of cells to say if they're significant or not, they'll give a percentage. So that's probably a really good question for a geneticist that's caring for it to look at that again and see what they think about whether there's mosaicism there. How often do women with Turner syndrome get multiple sclerosis? There's not a known connection with that, although that's something it sounds like would be really good to look at. That would be good information to collect, say, in the registry, and find out if that's something that's happening commonly. That's one great thing about the registry is that as women become adults, they can still participate in the registry and add some of that information. So hopefully more to come on that if we see that there's a trend. What is the arm abnormality? So that's called Madelung anomaly. So the arm anomaly is Madelung anomaly, and that's where this part of the arm is not developed quite as fully. So the head of the ulna, which is the bone right here, does not develop as more, and they sort of slip next to each other with a little bit of curvature. It doesn't happen really, really frequently in, say, the majority with Turner syndrome, but it can happen. Will ring X be covered in the next session? Yes, absolutely. We're going to talk about how rings form and why they happen, and also what that means. And can TS be hereditary in some cases where multiple family members can have it? So that's great. And great question. In general, Turner syndrome is, like we said, a random event, not inherited from generation to generation. There may be rare cases where that could happen. One, if somebody with Turner syndrome, say with mosaicism, becomes pregnant, then their child could have Turner syndrome. Also, if there's some type of rare, say, translocation with the X chromosome causing a partial deletion, but exchanged with another chromosome. But that would be extremely rare, but could happen. What is known about 45X, 47XXX? That's another example of a Turner syndrome karyotype that we don't think of as the first one or two. And we'll talk more about that in the next lecture as well, but there can be some differences in that from 45X, 46XX, and there's some different issues that can come up with that. So let's see. Is there any research advancement in epigenetics regarding gene expression in Turner syndrome? There are some people working on that, on epigenetic expression with Turner syndrome and other syndromes that look similar to Turner syndrome. There's one called Kabuki syndrome that's very similar as far as the X expression. So we think that there's genes, we talked about those 20,000 genes, so there's some other genes that affect expression of the X and vice versa. But we're learning more about that. We don't know as much as we'd like. Is XP22.3, are you able to tell which parent passed on Turner syndrome knowing it doesn't matter? So it doesn't really change. There's some evidence that perhaps sometimes if we're missing the X from dad or the X from mom, there can be some variations, but it doesn't really change how our treatment plan, so we don't typically test for it because it doesn't change what we do. Mm-hmm.
Follow up on that. Mm-hmm. I know you don't typically test for it, but could you, like, for example, if I knew I was exposed to something where I could potentially have an issue, like, doesn't matter, I agree, doesn't matter who it is, but like, just wanting to know if you wanted to or needed to test for that and determine.
Right. So it's possible to do more often under a research, if somebody's doing a research project, but like, it's not a routine thing, but it's possible to do it. Is Turner syndrome considered intersex? No, people with Turner syndrome are females. Can early intervention affect gene expression? So that's a really good question. It doesn't really, at this point, affect gene expression because we haven't really learned how to do gene therapy for Turner syndrome, for instance, to affect how those genes on the X are expressed. However, what we do know is that early intervention can help with growth, with finding cardiac abnormalities earlier, monitoring for hearing loss, all of those things to make sure that we're monitoring for those medical issues and catching them early so that they're treated better. Yes?
I have a question about kind of related to the intersex question. I have a friend from college who has Turner syndrome.
Right.
They are going through the process of hormone treatment for going through transgender changes.
Right.
Have you ever worked with someone who has expressed that interest to receive that treatment, and did you see how it ended up?
That's a great question. I do primarily pediatrics with Turner syndrome. I haven't specifically had that as part of my practice, but I'm certain there are other people out there with experience with that as well. How does Noonan syndrome compare to Turner syndrome? So that's a really good question. People with Noonan syndrome, which is a different syndrome, it's caused by a separate set of individual genes that can have single gene mutations rather than a chromosome change. But people with Noonan syndrome can look very similar to people with Turner syndrome. So sometimes if you're examining a baby, they can look very similar and you might end up testing for both if you're not sure. But the heart problems in Noonan syndrome are very different, so that's often a big clue. Okay, is XP22.33Q28 a karyotype, and what does that mean? So that's a great question. I would need to see the whole karyotype, so that's probably a piece of the karyotype. It could be a ring or an isochromosome. It'd be hard to tell. It sounds like it's probably a piece of the report related, but it could be something like a ring. And we talked a little bit, are you able to tell which parent passed on Turner syndrome? And we are able to, but like I said, it's not something we do as a routine testing. Okay, any other questions that we have? Anything else? Oh, yes.
Yeah, the person that asked about Ehlers-Danlos, my sister has forms of Ehlers-Danlos.
Okay.
And my mom has pulmonomorphins. So there's something in there that's maybe they're usually collagen problems, but there could be something in there that destabilizes the chromosome area.
Gotcha. So that's a really good question.
Yeah.
That's a really good question. So we don't know of an interaction between, say, the connective tissue disorders like Marfan and Ehlers-Danlos, but of course there's a lot we don't know. And we also know that people can have more than one genetic condition. Sometimes single gene variants can happen. Some people with Turner syndrome can have additional genetic conditions as well, like neurofibromatosis, for instance. So there could be a connection, but I never say never, and I know that we have a lot to learn about, kind of how those genes interact and are connected. So again, something we should keep an eye on and learn more about, for sure.
Also some other ones like Bloom disease or Bloom syndrome. To do with the sister chromosome swapping occurs at a lot higher of a rate, and it
Right.
makes more errors.
Right. So there are genetic conditions that can cause the chromosomes to kind of hyperswap. We can have exchanges between our chromosomes when cells are dividing, but there are some conditions where they sort of hyperswap. Turner syndrome itself doesn't cause that to happen, but again, if somebody has one of those hyperswap syndromes, they can make more mistakes later after birth. It can do that. Can pregnancy alter a woman's DNA to the point of causing autoimmune issues? That is a great question, and I don't think we know all the answers to that as geneticists. We know there are a lot of genes that can cause autoimmune issues, and sometimes when you have a lot of hormone changes, autoimmune disorders can kind of rev up and become apparent even when they weren't there before. So maybe that change in the blueprint was there, but pregnancy can rev it up as a possibility. So sometimes pregnancy can kind of change how our bodies understand the blueprint and how our bodies react to the blueprint. Is ring mosaicism or classic? Okay, so ring X can often be mosaic, where some cells have the ring and some don't. Also, sometimes you have a karyotype where all of the cells have the ring. So that sort of a variation occurs. So it's a little different from classic, which we think is 45X in all cells. We think of classic. So it's a little bit different from that. My mother was unable to get pregnant after me. Is that common? So that, again, is a good question. If mom had Turner syndrome, sometimes we find that if a mother has Turner syndrome and has a child with Turner syndrome, of course, there'd be more difficulty becoming pregnant. But I'm not aware of, say, a mother having infertility issues due to a child having Turner syndrome. But again, that's another thing that we are collecting information on in the registry of, what's happened during pregnancy or what's happened in the family history too. So any other questions that I can answer? Anybody else? Do autoimmune and Turner syndrome go hand in hand? Yes, absolutely. Turner syndrome individuals do have a higher risk for autoimmune issues, like hypothyroidism, Crohn's disease, and ulcerative colitis, for instance, celiac disease. Not as much rheumatoid arthritis, although that can happen. So it does increase the chances. It doesn't mean that those things are going to happen for everybody, but it does increase the chance. And there's certain karyotypes where we have a higher chance for developing those autoimmune disorders. And that's something, again, we'll talk in the next lecture a lot more about that. Yes? Okay. Sure. Okay. It would depend a little bit on what your previous karyotype was. If your previous karyotype was very straightforward, then you may not need to. At this point, it probably wouldn't change a lot of your medical care, but if it was something out of curiosity that you're interested in, it's something that you could ask your geneticist or primary care about and see if if it's not a clear karyotype, they could rerun. So, anybody else? Any questions? Great. Well, it's been a pleasure talking with you all, and we'll be back in just a few minutes to talk a little more about specific karyotypes, and I'll be around in case anybody has any individual questions or additional questions later.